Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the phagocytosis. So phagocytosis, why exactly the phagocytosis takes place? And if the neutrophils and the macrophages have the ability to phagocytose different foreign bodies, why don't they phagocytose their own body cells, neighboring body cells? So the answer is, to get phagocytosed, there must be 2 to 3 things. Number one, there must be loss of smooth surface of the cell. And secondly, there must be loss of protective layer on the cell. So normal body cells have a smooth surface and they have a protective layer on them. That's why normal cells are not phagocytosed. And if a normal cell is damaged, and there is a loss of its smooth surface or loss of its protective layer, then it will definitely be phagocytosed by neutrophils or macrophages. Okay, so foreign bodies have in foreign bodies there is loss of smooth surface and there is loss of protective layer. That's why they are phagocytosed. And the third very important point regarding phagocytosis is opsonization. So what exactly is this opsonization? Opsonization is basically the tagging of the foreign body with the complement protein so that the phagocytic cell or phagocytic membrane eats it up easily. Okay, so there is a foreign body. We want to tag it so that the macrophages come and eat it up. Okay, so this is known as opsonization. So it's basically the interaction of the foreign body with the antibody and the C3 complement protein. And this C3 complement protein basically marks or tags the foreign body that this foreign body should be eaten. And then the neutrophil or the macrophage eats this foreign body up. Okay? So opsonization is a process by which the pathogens such as bacteria or viruses are marked for destruction by the immune system. So this is marking of the bacteria or viruses so that they must be destroyed by the immune system. This is known as opsonization. So this is the macrophage and this is a foreign body. So first of all, this foreign bodies will be tagged by the antibody, and this antibody will be tagged by C3 complement protein, and then the macrophage will come and eat this whole combination up. Okay. So now there are 2 types of cells which can do phagocytosis. One is the neutrophils, and the other ones are the macrophages, as discussed previously. Now, the neutrophils can do phagocytosis in both the blood and the tissues, whereas macrophages can just do the phagocytosis in the tissues. Then, neutrophils can do phagocytosis of 3 to 20 bacteria, whereas the macrophages can phagocytose hundreds of bacteria. Okay, so macrophages are the main phagocytosing cell because they can phagocytose hundreds of bacteria, whereas neutrophils can just phagocytose 3 to 20 bacteria. Macrophage can not only phagocytose the bacteria, they can also phagocytose red blood cells and even the malarial parasites. So this is the significance of macrophages. Number one, they can eat a lot of bacteria, they can eat RBCs, the damaged RBCs, and also the malarial parasites. Now, neutrophils will remain in the tissues for a few days, whereas macrophages, the tissue macrophages, the monocyte, when they are converted into the tissue macrophages, they survive for months and months in the tissues. Okay? Now, let's move on. Now, how exactly is the phagocytosis being done? You know, when the macrophage or the neutrophil reach near the foreign body, they will form pseudopodia around them. And then the pseudopodia will fuse around the foreign body, making a phagosome. Okay? So this is what is going to happen. This is a foreign body, which has antibodies on it. Now this is a phagocytic cell. For instance, this is a macrophage. Now this macrophage will do endocytosis of this foreign body, or do phagocytosis of this foreign body. And this foreign body, when phagocytosed, it will be an endocytotic vesicle known as phagosome. So now a phagosome is formed. Now this phagosome will get attached with the lysosomes. Now the lysosome will empty all their enzymes into this phagosome, making this phagosome a phagolysosome. Now these enzymes of the lysosome will digest this foreign body. And after digestion of this foreign body, all of its debris, all of this microbial debris will be exocytosed. Okay, so whole story starts with the phagocytosis, and then the formation of a phagosome. Then the lysosome gets attached to the phagosome, forming it a phagolysosome, and finally the microbial debris will be exocytosed out of the cell. Okay, so this is the lysosome is attached to phagosome. A digestive vesicle is formed. Now inside the digestive vesicle, the phagolysosome, the digestive vesicle, the digestion takes place. Because lysosome has proteolytic enzymes, it has lipases, it will break down the proteins of the foreign body, it will break down the lipid of the foreign body. Okay, and it will digest it. The lysosomal enzymes will digest it, the foreign body. Now, other than the lysosome, peroxisomes can also be attached to the phagosome. Now, as you know, the peroxisomes have enzymes which basically kill by oxidation. So they have a lot of important enzymes which have huge ability to kill by oxidation. These enzymes include the superoxide, the hydrogen peroxide, the hydroxyl ions, and the hypochlorite ions. So the enzymes present in the peroxisomes are superoxide, hydrogen peroxide, hydroxyl ions, and hypochlorite ions. Okay, so this is phagocytosis, and these were the steps of phagocytosis. The important points to remember are the opsonization, which is basically the tagging, and then the macrophage have more ability of phagocytosis as compared to the neutrophils, and then when the phagocytosis is done, it gets attached with lysosomes and peroxisomes, which digest the foreign particle. So this is it. This was phagocytosis. See you in next part.